Hey guys, Tony here. What if I told you that there was something that could potentially cure stuff like cancer, AIDS, ALS? What if I told you that this thing is being developed? Can you guess what it is? It's CRISPR. Basically, what CRISPR is editing the DNA of any kind of cell to make them do whatever you want them to. So say a cell has a damaged or mutated DNA that's making them duplicate really rapidly. That's basically what cancer is. By replacing that mutated DNA with the DNA that you want, with a DNA that a scientist has created that's regular, that's normal, that won't do that, it'll stop doing it. That's why this technique, that's why CRISPR is so valuable, because it's relatively cheap, it's very accurate, and it has the potential to do so much. CRISPR doesn't have to be this big, grand, life-changing thing. Actually, there's a guy in Oakland named Josiah Zahner that runs a lab with a couple of his friends out of his own garage. He basically creates kits and teaches people how to use CRISPR and other gene editing techniques to make glowing beer. All right, let's go check it out.
Hey man, how's it going?
Good. Elbows. I got my gloves on.
So we are a startup and we're run out of a house in Oakland, California. And we took the whole bottom floor and built it into a genetic engineering lab. I worked at NASA for a few years in their synthetic biology program, engineering organisms so that we could use them to help people when they go to Mars or when they're in space a long time. CRISPR is a new genetic engineering technique that allows these genome edits to be done, except really simply, and it doesn't take a lot of time or money. What we're trying to do is give people the ability to genetically engineer something that they can use in their everyday life. So like modifying a yeast that they could then use for brewing or baking or something like that.
So if it's that easy, do you mind maybe doing a little experiment with me then?
Yeah, we could totally do an experiment. Do some genetic editing right here. Okay, so we're gonna do a CRISPR experiment. I'm gonna show you how it's done. Put on some gloves.
Okay, so the experiment ended up being really long, so I'm just going to explain this to you. You first scrape off a little bit of yeast and add it to a saline solution so everything mixes well. See? And then you add Cas9, which is a protein that's going to cleave off the DNA that you don't want and replace it with the one you do want. And then you add a gRNA, which is a guiding RNA that'll lead our Cas9 to where we want it to go. And then you add the template, which is what you want the DNA to look like after this is all finished. In this case, our new DNA is going to make protein that makes our yeast glow. It's the same one that makes jellyfish glow. That's so neat. And then you put the whole solution into a nice bath so it stays at a nice temperature. And then after a few days, you smear it onto a plate and see what happens.
See, this is the before and after. Whoa. I think the future of it is in people's hands. The technology really started to change everything when everybody got access to it. And they're doing things that I couldn't even imagine. After that, I think it's definitely going to be in the human genetic modification realm. There's a lot of talk about gene therapy and using CRISPR in medicine, but what about outside medicine? What about for cosmetic reasons? What about just if you're trying to get high or something?
All right, thanks guys, see you later.
See you later.
See ya. So I just got back to the office and it's so cool that even someone like me can make yeast glow. Who even knew that yeast can glow? It's really cool that with CRISPR on one hand, you have the potential to cure cancer, cure all these diseases. But on the other hand, you can just use it to do something as fun as make glowing beer. You don't have to be a scientist to utilize this technique and just make cool, fun things at home. All right guys, I'm out. I'll see you later. Hey Chewy, have you heard of CRISPR? No? No help.